Hello everyone, my name is Sonia. Today we are going to discuss about flow cytometry, principle and design of flow cytometer. Flow cytometry is a technique used to detect and measure physical and chemical characteristics of a population of cells or particles. Cyto means cell and metry means count or measurement. Principle of flow cytometry. Flow cytometry is a technique which enables rapid analysis of statistically significant number of cells at single cell level. The main principle of this technique is based on scattering of light and emission of fluorescence which occur when a laser beam hits the cells moving in a directed fluid stream. This is a diagram of flow cytometry. Next is flow cytometer. A flow cytometer is made up of three main systems: fluidics, optics and electronics. The fluidic system transports particles in a stream to the laser beam for interrogation. The optic system consists of lasers to illuminate the particles in the sample stream and optical filters to direct the resulting light signals to the appropriate detectors. The electronic system converts the detected light signals into electronic signals that can be processed by the computer. Fluidics. The fluidic system consists of flow cell, central channel and outer sheath. Central channel through which the sample is injected. Outer sheath contains faster flowing fluid known as sheath fluid, enclosing the central core. As the sheath fluid moves, it creates a massive drag effect on the narrowing central chamber. Under optimal conditions, the fluid in the central chamber will not mix with the sheath fluid. Hydrodynamic focusing produces a single file stream of particles. In this diagram, we can see sheath fluid and hydrodynamic focusing region. Here we can see cells in a single file. Next system is optics. After the cell delivery system, the need is to excite the cells using a light source. Lasers are more commonly used as they are highly coherent and uniform. Example, argon laser. The interrogation point. This is the place within the flow cytometer where the laser light hits the individual particles as they pass in front of the laser, one at a time. Light scattering in flow cytometer is mainly two types: forward scatter channel and side scatter channel. Forward scatter channel or FSE, the incident light scattered in forward direction is called forward scattered light. FSC is proportional to cell surface area or size. Side scatter channel, the incident light scattered at an angle of 90 degree. SSC is proportional to cell granularity or internal complexity. In this diagram, we can see forward scattering and side scattering. Emission of light. A fluorescent compound absorbs light energy over a range of wavelengths that is characteristic for that compound. This absorption of light causes an electron in the fluorescent compound to be raised to a higher energy level. The excited electron quickly decays to its ground state, emitting the excess energy as a photon of light. This transition of energy is called fluorescence. Filters in optics. Optical filters are designed such that they absorb or reflect some wavelengths of light while transmitting others. Types of filters: long pass, short pass and band pass. Detector in optics. The photodetector convert the photons to electrical impulses. Two common types of detectors used in flow cytometry: photodiode and photomultiplier tube. Electronics. The electronic system converts photons to photoelectrons. It measures the amplitude of the photoelectron's pulse and converts it into voltage pulse. The process involves signal shaping, amplification, and conversion from analog to digital format. The voltage pulse is assigned a digital value by analog to digital converter. All of the data associated with each individual cell are binned and stored in digital data file that can be read and analyzed using the appropriate analysis software. This is the schematic representation of electronic system. Data analysis. Flow cytometry data is typically represented in one of two ways: histograms, which measure or compare only a single parameter, and dot plots, which compare two or three parameters simultaneously on a 2 or 3-dimensional scatter plot. Basic mechanism of flow cytometer. First, taking biological sample, and next step is label it with a fluorescent marker. Third step is cells move in a linear stream through a focused light source. Next step is fluorescent molecule gets activated and emits light that is filtered and detected by sensitive light detectors. Next step is conversion of analog fluorescent signals to digital signals. Thank you.